Hey there, everybody, and welcome to this episode of Woodside Academy. Today we're going to be talking about mitosis and meiosis. These are types of cell division, one of them being division of body cells or somatic cells, and the other one being division or creation of sex cells or gametes. First, we're going to talk about mitosis. All right, this is basically your cell cycle, and in your cell cycle, right, we have interphase. And interphase is the first phase of mitosis, right? There are three phases to interphase. G1, which is a growth phase. This is when your cell is just starting to grow and get a little bit bigger. Then we have the S phase, or the synthesis of DNA. So here we start to replicate all of the DNA that's inside of the nucleus. And then we have the G2 phase, or a second growth phase. So this is 90% of the cell cycle right in here, right? G1, S, and G2. And then we actually enter the mitosis phase of the cell cycle, where we have the actual cell division. Right here we have prophase, metaphase, anaphase, telophase, and cytokinesis. And all of these together, these four, prophase, metaphase, anaphase, and telophase, make up what we call mitosis, right? And this is, again, cell division of body cells. So I like to remember this as mitosis occurs in mitoses, right? Okay, there is no real dividing going on during interphase. Okay, so, that is what's happening there. At the end of mitosis is when we actually do the cell division. This is called cytokinesis, where the entire cell divides into two. In animal cells, you just have a pinching off of the cell membrane, right? And in plant cells, you actually have a cell plate that starts to come between the two and eventually forms the cell wall. Right, so that's what's happening at the end of mitosis, going into the next phase of interphase. The only time that you can really see chromosomes, so they are actually visible in human cells, is during mitosis. During the 90% of cell cycle, or cell's life, you cannot see chromosomes. They're condensed around histones as a material called chromatin. But during mitosis, all your chromatin, all your chromosomes actually condense so you can see them, and they look like little X's, like you see right here. In the middle, they're connected by a centromere, and then you have the chromatids that are off to the side. So you only see those during prophase, metaphase, anaphase, and telophase. So what are the phases of mitosis? Well, 90% of it is interphase. Remember that for the cell cycle. Okay, so here you do not see the chromosomes, right? They're condensed, or actually are uncondensed, and so they just look like a blob. Now during prophase, the P cycle, you can see the chromosomes are now condensed, and they look like actual material. In metaphase, right, they're all lined up across the middle, and so they're lined up across the middle of the cell, or the equators. Off to the sides, you can think of it as the North and South Pole, because we have centromeres that sit off there, and or centrioles that sit off to the side and actually draw these chromosomes along spindle fibers to get to the ends. All right. After metaphase comes anaphase, and during this phase, the chromosomes start to ride along these spindle fibers out towards the edge of the cell where the centrioles are. So the centrioles put out the spindle fibers that act like cables, sort of like a ski car or something. And they take this lift out to the end, and they'll separate that way. Telophase is just a little more, uh, a further continuation of anaphase. The chromosomes are really getting out to the poles, and you can really see how they're separated, and they're really starting to cluster into two different groups. And then we get into cytokinesis, which we talked about before, and now you have your actual division of your two cells, the chromosomes decondense, and now you're back to chromatin in the nucleus. So that's what's happening during mitosis. Meiosis is a little bit different. Now, meiosis is formation of sex cells, right? So what you have going on here is you do have two phases to meiosis, actually. You have meiosis 1, which is essentially mitosis, right? And during meiosis 1, you will go ahead and replicate everything, right? And then during meiosis two, you'll just divide. So meiosis one is a replication and division, while meiosis two is just division. So here we're moving through, we replicate everything and we get all lined up like we do in metaphase, right? And then we have what's called homologous pairs splitting. So you have basically pairs from your mother and pairs from your father, and they split up, and then they come down here into the second phase, and they're lined up again in the middle. This was metaphase 2 and metaphase 2, and then they will just divide, and there's no replication. So you wind up with 4 cells that are each half of the original chromosome number. And that's really what happens in meiosis. All right? So when I talk about homologous pairs of chromosomes, what we're looking at is that we have these sister chromatids, right, that are here, and they will come together and pair up. These are chromosomes, one from the father, one from the mother, that all represent basically similar traits, or traits for the same parts. So you have the sister chromatids, and they're next to each other, and when they pair up, they make a tetrad, and this happens during prophase of meiosis 1. Now, during this, we're going to see something that's called crossing over, and this really helps to increase, right, to increase our genetic variation, right? So here's crossing over. So during prophase 1, right, which is part of meiosis, the first phase of meiosis, you get your paternal chromosomes and your maternal chromosomes, and they form your tetrad, and they are lined up next to each other, and you can see how they switch here. A part of the maternal chromosome, the red, gets over here in the orange, and part of the paternal chromosome gets switched over to the mothers. And then you wind up with this good variation. So this is good for us. We want variation in our genes because it helps with keeping us healthy, right? And it's good heterogeneity, right? So we get to find the best and the most fit genes, and that's what we're looking for there. A little bit of Darwinism, okay? And here's another example of what's going on. You can see here that you would have A and B, right, from the green chromosome being switched with A and B from the white and yellow chromosome. So again, it generates diversity, and it occurs in prophase of meiosis 1. Right, so there you have the two that were switched. Now, how can we compare these things? This is good to know because sometimes you might get asked a little question about this or have to look at a chart or just be able to recognize stuff. Now, mitosis, again, is somatic cells or body cells. And so this is division of skin cells and liver cells and bone cells and brain cells, right? While meiosis is formation of gametes, your sperm and your eggs, right? So mitosis has 4 phases to it: prophase, metaphase, anaphase, and telophase, while meiosis has 8 and has prophase, metaphase, and anaphase, and telophase 1. And here you have growth and division, replication and division. Then you have a second set, prophase, metaphase, anaphase 2, where you only divide. It's called reduction division, right? How about numbers of cells that you have that are formed? Well, in mitosis, you have 2 diploid cells. Diploid, or 2N, means this full chromosome number. So all of the chromosomes that were represented in the parent cell are still represented in the daughter cells. Versus meiosis, where you wind up with 4 haploid cells, each of the cells having half of the chromosome material that the original parent had. Now remember, we had to replicate and then divide the first time around. So the second time we just divided, and that's how we wind up with 4 cells, each with half of the information. So with males we get 4 sperms, and with females you get 4 eggs, and only one of those is viable. So what are we looking at here versus identical or anything like that? For mitosis, you have 2 identical twin cells. For meiosis, you have 4 different cells, and that's where we get our genetic combination from. Again, here's another chart just kind of looking at it. How many divisions do we have? All right, 1 division in mitosis, 2 divisions in meiosis. Number of daughter cells, 2 versus 4. And daughter cells are the ones that are produced at the end. How about genetically identical? The ones in mitosis are, right? Because mitosis occurs in mitoses, you want all of those cells to look the same. and then they are not identical in meiosis. Right? That's why you and your sister or brother or cousin do not look the same. All right? Chromosome number, right? In mitosis, they are the same as a parent, and in meiosis, they are different. All right? So that's what you were looking at in that case. All right, so here's a graphic that's comparing the two. So sometimes you like to look at it. All right, we first talked about mitosis, so let's look at that. It's on the right. Here's your cell, okay, and you are now in prophase, because remember, interphase is not actually a part of cell division. It's part of the cell cycle, but not cell division. So we're starting out with prophase here, where you see all of the chromosomes, and then you come down, right, and you're lined up in metaphase. Now we're going to go through anaphase, and then eventually to telophase, and then cytokinesis, and we finish up with two identical cells. And then over in meiosis, we start with the cell, we replicate everything and then divide everything. Right? So this section is basically like mitosis. And this is where crossing over can occur also. Then we come into the second phase where we line up again during through my metaphase, and then just divide. So here we have reduction, division. And that's all for today. And thank you very much. If you have uh, any questions about this, just go back and look at it again, or feel free to look it up on YouTube and find some links and to supplement your understanding. All right, thank you and have a great day.